Hello, my name is Naomi Pineda, and today I will be talking about ion channels and neuronal communication. Ion channels are specialized proteins found in the cell membranes of neurons and other cells. They play a crucial role in neuronal communication, which is the process by which neurons send and receive electrical signals. Neuronal communication involves the generation and propagation of electrical impulses called action potentials. These action potentials allow information to be transmitted rapidly across long distances within the nervous system. Ion channels are responsible for the flow of ions such as sodium, potassium, calcium, and chloride across the cell membrane. They regulate the electrical properties of neurons by controlling the movement of ions in response to various stimuli. Voltage-gated ion channels are particularly important in the generation and propagation of action potentials. They respond to changes in membrane potential and open or close accordingly, allowing the rapid movement of ions in response to electrical signals. The precise regulation of ion channels is critical for proper neuronal communication. Different types of ion channels are expressed in specific regions of the neuron, enabling precise control over the timing and amplitude of electrical signals. Dysregulation or dysfunction of ion channels can lead to impaired neuronal communication, resulting in various neurological disorders. Neurons are specialized cells that form the building blocks of the nervous system. They play a vital role in transmitting information throughout the body and are responsible for many essential functions, including sensory perception, motor control, and cognitive processes. The basic structure of a neuron consists of three main parts: the cell body, dendrites, and the axon. The cell body contains the nucleus and most of the cellular machinery, while dendrites receive incoming signals from other neurons or sensory receptors. The axon is a long, slender projection that transmits electrical impulses, known as action potentials, away from the cell body. Neurons communicate with each other through a process called synaptic transmission. When an electrical signal known as an action potential reaches the end of an axon, it triggers the release of chemical messengers called neurotransmitters into the synapse, which is the junction between the two neurons, initiating a new electrical signal in the receiving neuron. The action potential is the basis of neuronal communication. It is an all-or-nothing electrical impulse that is generated when a neuron receives a strong enough stimulus. This stimulus causes a temporary change in the electrical charge across the neuron's membrane, known as depolarization. If the depolarization reaches a certain threshold, voltage-gated ion channels in the membrane open, allowing an influx of positively charged ions, such as sodium, into the neuron. This rapid influx of positive ions can cause a rapid change in the electrical potential of the neuron, creating an action potential. Once an action potential is initiated, it propagates along the axon, allowing the electrical signal to travel over long distances. This propagation occurs through a process called saltatory conduction, where the action potential jumps from one node of Ranvier to the next, speeding up the transmission of the signal. The action potential allows neurons to transmit information rapidly and efficiently, ensuring the proper function of the nervous system. It is the basis of various complex processes, including sensory perception, motor control, learning, and memory. By modulating the frequency and pattern of action potentials, neurons can encode and transmit information across vast networks, enabling the coordination and integration of signals throughout the body. Ion channels are integral membrane proteins that play a fundamental role in the electrical signaling and communication within cells. They are responsible for regulating the flow of ions across the cell membranes, thereby influencing various physiological processes such as nerve conduction, muscle contraction, and hormone secretion. The structure of ion channels consists of membrane-spanning regions and an ion-selective pore. The membrane-spanning regions are hydrophobic segments of the protein that traverse the lipid bilayer of the cell membrane. These regions anchor the ion channel in the membrane, allowing it to span the entire thickness of the membrane. Typically, multiple membrane-spanning segments are present in an ion channel, forming transmembrane domains. The ion-selective pore is a central feature of ion channels. It is formed by a specific arrangement of amino acids within the transmembrane domains, creating a channel or pore through which ions can pass. The pore is often lined with amino acids that possess charges or polar groups, enabling them to interact selectively with specific ions. This selective interaction determines the ion specificity of channel, allowing it to conduct specific types of ions while excluding others. The ion channel's pore can adopt different transformations, including open and closed states, which regulate ion flow. In the closed state, the pore is blocked or constricted, preventing ion passage. In the open state, the pore is unobstructed, allowing ions to move across the membrane. The opening and closing of the pore are controlled by various mechanisms such as changes in voltage, binding of ligand, or mechanical forces. The structure of ion channels is diverse and can vary significantly depending on the specific type of channel and the ions it conducts. Some ion channels consist of a single protein subunit, while others are composed of multiple subunits that come together to form a complex. The assembly of subunits can contribute to the function, properties, and regulation of ion channels. Now let's go over the different types of ion channels. Let's start with ligand-gated ion channels. These are activated or inhibited by the binding of specific chemical messengers known as ligands to the receptor site of the channel. These ligands can be neurotransmitters, hormones, or other signaling molecules. When the ligand binds to the receptor, the ion channel undergoes a conformational change, leading to the opening or closing of the ion channel pore. This allows the selective passage of ions depending on the specific receptor and ligand involved. Ligand-gated ion channels are crucial for fast synaptic transmission in the nervous system and are involved in various physiological processes, including sensory perception and neurotransmitter release. So let's move on to mechanically gated ion channels. These respond to the mechanical forces or physical deformations of the cell membrane. They are typically found in sensory cells, such as those responsible for touch, hearing, and balance. When the cell membrane is mechanically stimulated, these ion channels open or close, allowing ions to flow across the membrane. This initiates electrical signals that are transmitted to the central nervous system for processing. Mechanically gated ion channels are critical for translating mechanical stimuli into electrical signals and play an essential role in sensory perception and mechanotransduction. So the next one would be always open ion channels. Unlike the other three types, always open ion channels are also known as leak channels. They are not regulated by any changes in voltage, ligands, or mechanical forces. Instead, they are constitutively open, allowing ions to continuously flow across the cell membrane. Leak channels contribute to the passive movement of ions down their concentration gradients, helping to establish and maintain the resting membrane potential of cells. This resting potential is crucial for proper cellular function and serves the baseline electrical state of the cell when it's not actively generating or receiving signals. Now let's move on to the last one, which would be voltage-gated ion channels. These are activated or deactivated by changes in the electrical potential across the cell membrane. They are responsible for generating and propagating action potentials in excitable cells, such as neurons and muscle cells. Voltage-gated ion channels have a specific threshold voltage that triggers their opening, allowing ions such as sodium, potassium, and calcium to flow through the pore. This generates electrical currents, enabling the transmission of signals over long distances. Ion channels play a crucial role in regulating ion flow across the cell membrane, allowing the controlled movement of ions in and out of cells. This regulation is vital for maintaining ion homeostasis, establishing electrical potentials, and facilitating various physiological processes. Here are some key roles of ion channels in regulating ion flow. First, establishing ion concentration gradients. Ion channels contribute to the establishment and maintenance of concentration gradients across the cell membrane. These gradients are critical for cellular processes such as osmosis, nutrient uptake, and cell signaling. Ion channels selectively permit the movement of specific ions such as sodium, potassium, calcium, and chloride, enabling ions to move from areas of high concentration to low concentration. Now let's move on to controlling resting membrane potential. Ion channels, particularly leak channels, influence the resting membrane potential of cells. Leak channels allow a small and continuous flow of ions across the membrane, contributing to the polarization of the cell. This resting membrane potential provides the baseline electrical state of the cell when it is not actively transmitting or receiving signals. It plays a crucial role in processes such as nerve conduction and muscle contraction. Generating and propagating action potentials. Voltage-gated ion channels are responsible for the generation and propagation of action potentials, which are brief electrical impulses that allow long-distance communication within the nervous system. When a cell depolarizes beyond a certain threshold, voltage-gated ion channels open, causing an influx or efflux of ions, triggering an action potential. This electrical signal is propagated along the cell membrane, allowing for rapid transmission of information. Mediating synaptic transmission. Ion channels, particularly ligand-gated ion channels, are essential for synaptic transmission, the process by which neurons communicate with each other. When an action potential reaches the presynaptic terminal, it triggers the release of neurotransmitters. These neurotransmitters bind to a ligand-gated ion channels on the postsynaptic membrane, leading to the opening or closing of ion channels. This allows the flow of specific ions, altering the membrane potential and transmitting the signal from one neuron to the next. Facilitating sensory perception. Ion channels, including mechanically gated ion channels, play a significant role in sensory perception. For example, in sensory cells involved in touch, hearing, and vision, mechanically gated ion channels respond to physical stimuli and convert them into electrical signals. This allows sensory information to be transmitted to the central nervous system for processing and interpretation. The last one would be regulating cellular processes. Ion channels play a role in regulating various cellular processes, including cell volume, pH balance, hormone secretion, and cell proliferation. By controlling the movement of ions across the membrane, ion channels help maintain the optimal cellular environment and support proper cell function. Sodium and potassium channels play a crucial role in the generation and propagation of action potentials. The electrical signals responsible for nerve conduction and communication within the nervous system. The differences in ion selectivity and gating properties of these channels contribute to their distinct functions in action potential generation. So let's start with sodium channels. Sodium channels are primarily responsible for the rapid depolarization phase of the action potential. They have high selectivity of sodium ions and open in response to membrane depolarization. Sodium channels exist in different states: closed, open, and inactivated. At resting membrane potential, most sodium channels are closed. However, when the membrane is depolarized beyond a specific threshold, the channels undergo a conformational change and rapidly open, allowing an influx of sodium ions. This influx of positive charge further depolarizes the membrane, leading to the rising phase of the action potential. Sodium channels then quickly undergo inactivation, becoming refractory to further depolarization, which is crucial for the propagation of action potentials in one direction along the axon. Now let's move on to potassium ion channels. Potassium channels are involved in the depolarization and restoration of the resting membrane potential during an action potential. They have higher selectivity for potassium ions and exhibit different gating properties compared to sodium channels. Potassium channels open more slowly than sodium channels and contribute to the delayed polarization phase of the action potential. As the membrane depolarizes, potassium channels gradually open, allowing the efflux of potassium ions, which repolarizes the membrane by restoring the negative resting membrane potential. The opening of potassium channels also helps in terminating the action potential and preparing the neurons for subsequent firing. The differences in ion selectivity and gating properties between sodium and potassium channels are critical for the proper generation and propagation of action potentials. The rapid opening of sodium channels and subsequent influx of sodium ions during depolarization lead to the initiation of the action potential, while slower opening of potassium channels and efflux of potassium ions allow for repolarization and restoration of the resting membrane potential. The distinct functions of sodium and potassium channels are essential for the effective and efficient transmission of electrical signals along neurons. Their coordinated activation and inactivation ensure the propagation of action potentials in one direction and enable precise and reliable communication within the nervous system. It is important to note that other types of ion channels, such as calcium channels, also play significant roles in action potential generation and synaptic transmission. The coordinated interplay between different types of ion channels contribute to the complex dynamics of neuronal excitability and information processing in the nervous system. Calcium channels play a crucial role in synaptic transmission by mediating the influx of calcium ions into the presynaptic terminal. The influx of calcium triggers the release of neurotransmitters from the presynaptic neuron into the synaptic cleft, leading to the transmission of signals of the postsynaptic neuron. When an action potential reaches the presynaptic terminal, it depolarizes the membrane and activates voltage-gated calcium channels. These channels are present on the presynaptic membrane and are specifically referred to as the N-type calcium channels, PQ-type calcium channels, or L-type calcium channels. Activation of these channels allow calcium channel ions to flow into the presynaptic terminal down the electrochemical gradient. The entry of calcium ions into the presynaptic terminal has several important consequences. Firstly, calcium ions bind to proteins known as synaptotagmines, which are located on the surface of synaptic vesicles containing neurotransmitters. This binding event triggers a conformational change in the synaptotagmines, leading to the fusion of the synaptic vesicles with the presynaptic membrane and subsequent release of neurotransmitters into the synaptic cleft. Secondly, calcium ions also modulate the machinery responsible for docking and priming of synaptic vesicles, ensuring their proper release. Calcium influx promotes the interaction of synaptic vesicle proteins with proteins present on the presynaptic membrane, enabling the fusion process. Neurotransmitters are chemical messengers that transmit signals across the synapse. Once released into the synaptic cleft, neurotransmitters diffuse across the cleft and bind to the specific receptors located on the postsynaptic membrane. The binding of neurotransmitters to their receptors initiates a series of events that ultimately leads to the generation of a postsynaptic potential, either excitatory or inhibitory, depending on the type of neurotransmitter and receptor involved. After the neurotransmitters have completed their role in transmitting the signal, they are either taken back up into the presynaptic terminal through the reuptake mechanism or enzymatically degraded to degraded to terminate their action. Channelopathies refer to a group of disorders that are caused by mutations or dysregulation in ion channels. These disorders can affect various organ systems, including the nervous system, in the context of neuronal communication. Channelopathies disrupt the normal functioning of ion channels, leading to impaired electrical signaling and neurotransmission. Some examples include episodic ataxias, which is a group of genetic disorders characterized by episodic imbalance and coordination problems. Mutations in potassium channel genes such as KCNA1 and CACNA1A can lead to episodic attacks of ataxia, which are brief periods of uncoordinated movement. The long QT syndrome is a cardiac channelopathy that can also affect neuronal excitability. It is caused by mutations in potassium or sodium channel genes, leading to prolonged repolarization of cardiac cells and an increased risk of life-threatening arrhythmias. In some cases, individuals with this disease may experience seizures and syncopal episodes due to abnormal neuronal electrical activity. Hypokalemic periodic paralysis is a disorder characterized by episodes of muscle weakness or paralysis, often triggered by strenuous exercise, fasting, or high carbohydrate meals. It is caused by mutations in voltage-gated calcium channels or sodium channels, resulting in abnormal ion influx, impaired muscle cell excitability. Myotonia congenita is a neuromuscular disorder characterized by delayed muscle relaxation after contraction, causing muscle stiffness or myotonia. It is associated with mutations in chloride channel genes such as CLCN1, which lead to impaired chloride ion conductance and altered muscle cell excitability. Last, epilepsy. Some forms of epilepsy are attributed to channelopathies. For example, mutations in sodium channel genes, including SCN1A and SCN2A, have been identified in certain forms of epilepsy, such as Dravet syndrome and benign familial neonatal infantile seizures. These mutations disrupt normal sodium ion flow, leading to increased neuronal excitability and susceptibility to seizures. These examples illustrate the impact of ion channel disorders on neuronal communication. Dysfunctional ion channels can disrupt the delicate balance of ion concentrations and alter the electrical properties of neurons, affecting their ability to generate and transmit signals accurately. Understanding channelopathies is crucial for unraveling the underlying mechanisms of these disorders and developing targeted therapies to restore normal ion channel functions and improve neuronal communications. Now let's move on to neurological disorders and ion channels. Let's start with Parkinson's disease, which is a neurodegenerative disorder characterized by the progressive loss of dopamine-producing neurons in a brain region called the substantia nigra. Dopamine is a neurotransmitter that plays a crucial role in regulating movement, reward, and motivation. The loss of dopamine-producing neurons leads to a significant reduction in dopamine levels in the brain, resulting in the motor symptoms associated with Parkinson's disease, such as tremors, rigidity, and slowness of movement. Dopamine channels, specifically dopamine receptors, are involved in the transmission of signals mediated by dopamine. There are several subtypes of dopamine receptors classified D1 like receptors, D2 like receptors. These receptors are G protein coupled receptors located in the postsynaptic membrane of neurons that respond to dopamine binding. Now, in Parkinson's disease, the loss of dopamine producing neurons results in a reduced stimulation of dopamine receptors, particularly the D2-like receptors. The D2-like receptors primarily exert inhibitory effects of the basal ganglia, a group of structures involved in motor control. With a reduced activation of D2-like receptors, there is an imbalance between the inhibitory and excitatory signals in the basal ganglia circuitry, leading to abnormal motor function. The standard pharmacological treatment for Parkinson's disease involves the administration of levodopa, a precursor to dopamine. This is converted into dopamine in the brain and helps compensate for the dopamine deficiency. Additionally, medications known as dopamine agonists can directly stimulate dopamine receptors, bypassing the need for dopamine production. By targeting dopamine receptors, these medications aim to restore the balance between inhibitory and excitatory signals in the basal ganglia and alleviate the motor symptoms such as Parkinson's disease. Epilepsy and potassium channels. So, epilepsy is a neurological disorder characterized by recurrent seizures, which are abnormal electrical discharges in the brain. These seizures can manifest as altered consciousness, convulsions, sensory disturbances, or abnormal behaviors. Potassium channels are critical regulators of neuronal excitability and play a key role in preventing excessive neuronal firing. Potassium channels are responsible for controlling the efflux of potassium ions out of neurons, which help maintain the resting membrane potential and repolarize the cell after an action potential. Dysfunction or dysregulation of potassium channels can disrupt the balance of potassium ions and alter the electrical excitability of neurons, potentially leading to hyperexcitability and seizures. Various forms of epilepsy have been associated with mutations in genes encoding potassium channels. For example, mutations in the KCNQ2 and KCNQ3 genes, which encode 4 subunits of specific types of potassium channels called the M channel, have been linked to benign familial neonatal seizures and benign familial infantile seizures. Additionally, mutations in other potassium ion channel genes such as KCNQ1 and KCNT1 and KCNA1 have been identified in different types of epilepsy syndromes, including the early onset epileptic encephalopathies. Alterations in potassium channels function can lead to changes in neuronal excitability, making neurons more prone to generating abnormal electrical discharges and seizures. Understanding the role of potassium channels in epilepsy helps shed light to the underlying mechanisms of the disorder and may contribute to the development of targeted treatments aimed at restoring normal potassium channel functions and reducing seizure activity. Drugs that target ion channels play a significant role in the treatment of various medical conditions. These medications are designed to modulate the activity of specific ion channels, either by enhancing or inhibiting their function. First, we have calcium channel blockers, sodium channel blockers, potassium channel blockers, GABA receptor agonists, and methyl D-aspartate receptor antagonists, chloride channel activation or inhibitors. In conclusion, ion channels are essential components of neuronal communication, enabling transmission of electrical signals within the nervous system. They regulate the flow of ions across membranes, playing a critical role in generation and propagation of action potentials, as well as the release of neurotransmitters at synapses. The precise control of ion channels activity is crucial for maintaining the balance between excitatory and inhibitory signals in neurons. Dysregulation or dysfunction of ion channels can disrupt this delicate balance, leading to impaired neuronal communication and the development of various neurological disorders. The study of ion channels has provided invaluable insights into the fundamental mechanism of underlying neuronal functions and has paved the way for the development of targeted therapies for neurological conditions. Drugs that modulate ion channel activity have revolutionized the treatment of diseases such as epilepsy. Advancement in our understanding of ion channels continue to expand, with ongoing research exploring their roles in neurodegenerative disorders, psychiatric conditions, and sensory processing. By unraveling the complex interactions and functions of ion channels, scientists and clinicians aim to develop innovative therapies that can restore proper neuronal communication and improve the lives of individuals affected by neurological disorders. Here is a list of my references, and thank you so much for listening.